So nice to meet you, everybody, and it's an honor to me to present Dr. Professor Lidija Klampfer from Department of Oncology in Albert Einstein College of Medicine in New York. That here today she will speak about her special field of interest, namely tumor microenvironment and tumor progression. Lidija Klampfer has been educated in Ljubljana at University of Medicine, a School of Medicine and Biotechnology Faculty since 1985. And she is actually a member of Albert Einstein Cancer Center in New York. She also joined New York Memorial Sloan Kettering Institute as postdoc doctoral fellow in 1999, and also before in 1993 was visiting scientist at New York University. She got a lot of honors and awards, and she is referee for many research projects and scientific articles, and her literature work is quite extensive, and especially she is an expert, a recognized expert in the field of cytokines and inflammatory and colon cancer. So I will thank you a lot for to be here, and I invite you here to join this presentation and to introduce us your field.
Hvala lepa. Dober večer vsem. Jaz bom vas pozdravila v slovenščini, ker to je moj jezik. So me pa prosili, da predavam angleščini, ker menda imamo goste iz tujine. So welcome everybody. It's lovely to be here. It's a great place. One would want to talk about art, not about cancer actually, in this hall, but that's what I work on. As you all know, cancer is a disease of genes. The progression from a normal to malignant cells requires changes in DNA. It occurs in steps, and drivers of oncogenic transformation are mutations in oncogenes. These are genes that promote tumor growth, and the other class of genes that are frequently mutated in cancers are tumor suppressor genes. These are genes whose normal role is to suppress growth of cancer cells. Colon cancer develop in at least 7 steps, but probably many more. What is important is that the first step in transformation of intestinal epithelial cells would always be loss of tumor suppressor gene called APC. And that will be important for my talk because the inactivation of this tumor suppressor gene induces Wnt signaling in cancer cells. In the past 10 years or so, it's become clear that the speed of this transition from normal cells to malignant cells is not determined just by genetic changes that occur in the cell, but it's influenced by the tumor microenvironment. A tumor-supportive microenvironment would speed up this process. A tumor-suppressive microenvironment could, if we are lucky, arrest tumor progression at the stage of adenoma, or would at least prevent the metastasis, which is severe complication of cancer, as we all know. So in my lab, we are studying how tumor cells talk to the cells that are present in the tumor microenvironment. Tumor microenvironment is a complicated structure. It's populated by a variety of cells. This is how I indicate macrophages. You find T cells, you find B cells, neutrophils, mast cells. An important component of the tumor microenvironment is extracellular matrix. And the question actually I always get when I talk about my work is that some of these cells, macrophages in particular, actually have inherent ability to destroy tumor cells. But it looks like that in established tumors, tumor cells have learned how to make the cells in tumor microenvironment work for them. And the way cells in the tumor microenvironment affect the progression of tumors is by secreting a variety of soluble factors which affect growth and survival of tumor cells. And pro-inflammatory nature of this tumor microenvironment actually is, in most cases, pro-tumorigenic. Soluble factors secreted by tumor cells bind to receptors on soluble factors secreted by cells in the tumor microenvironment, bind to the receptor expressed on tumor cells, and that results in activation of transcription factor and their translocation from the cytoplasm to the nucleus. Transcription factors are DNA binding protein. They bind DNA in sequence-specific manner. They bind to specific region in DNA. And they fall into two categories. They can activate transcription. In this case, a new protein will be made in the cell. Transcription factor can inhibit gene expression, and protein will be downregulated in the cell. I told you that the Wnt signaling is a signature oncogenic signaling in colon cancer cells. It truly is a complicated signaling, but it follows the basic rule. The ligand, in this case, is Wnt. The receptors are frizzled protein. But really a central player in Wnt signaling is beta-catenin. Beta-catenin is a normal, healthy cell kept in the complex that contains at least 20 protein. One of these protein, if you note, is the APC. That's the tumor suppressor gene, which is mutated in colon cancer. And the function of APC, or the function of the kinase, I'll be talking a little bit about this kinase, JSK3 beta, is actually to phosphorylate beta-catenin and to cause its degradation. When it's degraded, beta-catenin actually stays, protein stays in the cytoplasm. But when the APC is inactivated, which happens in colon cancer actually, beta-catenin is stabilized. It's translocated to the nucleus and regulate the expression of target genes, which are usually growth-promoting genes. CMYC is a typical example. An interesting thing is, if you take a look at the primary colon cancer cell, you note that beta-catenin is indeed nuclear, but it's nuclear only at the edge of the tumor. I don't know how well you can see that, but here I show you schematically. Beta-catenin is in yellow. Beta-catenin, nuclear localization at the edge of the tumor, and it's cytoplasmic in the nucleus, suggesting actually that there are factors in the tumor microenvironment that regulate beta-catenin transcriptional activity. And my story today will be actually to tell you that macrophages secrete factors that are important regulators of beta-catenin transcriptional activity. To get insight into crosstalk between tumor cells and macrophages, we did actually a screening procedure. We transfected tumor cell derived from colorectal cancer patient called HCT116 with a variety of gene reporters that we linked to the luciferase. These are gene reporters, these are artificial genes that actually represent DNA target sequence for DNA binding protein, and we established cells that express NF-kappa B, Wnt signaling reporter, AP1, Notch, STAT1, and STAT3. And what we did is we cultured these cells alone or in the presence of macrophages. The idea being, if macrophages secrete factors that regulate this pathway, the readout would be different when cells are cultured alone or in the presence of stromal cells. And when we culture the panel of this established cell line with macrophages, we noted that it's indeed only Wnt signaling that is activated by the presence of macrophages. Here is a little bit of the data. You can see that the addition of peripheral blood monocytes, which are the actual precursors of tumor-associated macrophages, or the presence of transformed monocyte, THP1 cells, increases Wnt signaling. More importantly, we showed that the expression of Wnt target genes is induced and that the proliferation of cells is increased when we culture tumor cells in the presence of macrophages. Although this experiment actually were done when we culture cells in direct contact, we later actually developed a system in the lab where we separated tumor cells from macrophages with the membrane that does not allow transmigration of cells, but allows exchange of soluble factors. And we saw that macrophages promote Wnt signaling through a soluble factor. The direct contact was not required for that. And when we examine what is present, what the cells secrete to the supernatant, we found that indeed when we co-culture tumor cells with macrophages, a variety of soluble factors were produced, a lot of cytokines and a lot of chemokines, CCL5, CCL3, and interleukin-1 beta. And these are all growth factors that could have an important role in biology of intestinal epithelial cells. Interleukin-1 beta was quite interesting for us. It's a marker of activated macrophages, and it can regulate the immune response, of course. We confirm by ELISA that interleukin-1 is produced by macrophages that are stimulated with tumor cells. Tumor cells alone did not produce interleukin-1. Macrophages alone did not, but when we co-cultured the cells, there was a good response in interleukin-1 beta. Tumor-associated macrophages have several unique characteristics. One of the characteristics of the cells is that they have active STAT1 signaling. STAT1 is a transcription factor, again, a DNA binding protein that is involved in signaling by many cytokines. And we noted that our transformed THP1 cells indeed have activated STAT1. So we asked the question whether STAT1 plays a role in the crosstalk between macrophages and tumor cells. And in order to answer this question, we had to get rid of STAT1 from macrophages. And we did that through small interfering RNA. It's a rather simple methodology. Small interfering RNAs are really very short, 20 nucleotide long duplexes of RNA that get incorporated into RISC. RISC stands for RNA-induced silencing complex. And when incorporated, they unwind. The antisense strand of RNA gets degraded. And RISC then guides, if you will, the remaining strand to your mRNA target. So we designed siRNA that is specific for STAT1. And as you can see here, we were quite successful in getting rid of STAT1 from macrophages. These are macrophages transfected by non-targeting siRNA. These are macrophages transfected with STAT1 RNA. And you can see that these macrophages actually are indeed devoid of STAT1. And we showed that such macrophages actually do not produce interleukin-1 at all. Maybe we can just concentrate on this first 3 bars. Tumor cells, no interleukin-1. When macrophages are transfected with non-targeting siRNA, good response, but no IL-1 whatsoever when cells are lacking this transcription factor. To confirm that IL-1 is indeed a factor that plays a role in Wnt signaling, we showed that recombinant interleukin-1 actually is quite sufficient to induce Wnt signaling, was sufficient to induce growth of tumor cells, was sufficient to inactivate JSK3 beta. If you remember the first slide, this is the kinase that is responsible for rapid degradation of beta-catenin. And interleukin-1 was sufficient actually to induce Wnt target gene in cells, such as c-Myc. c-Myc is a gene, when overexpressed, would drive cell proliferation, so is quite a potent oncogene. Not only was interleukin-1 sufficient to induce Wnt signaling, interleukin-1 also was required, actually, for macrophage-induced Wnt signaling. If we, through siRNA approach again, silence interleukin-1 in macrophages, such macrophages did not affect growth of tumor cells. Wnt signaling was not induced. Or if we use neutralizing antibodies specific to interleukin-1, these macrophages did not induce Wnt signaling. They did not induce clonogenic growth of tumor cells. Now, from a therapeutic point of view, siRNA is not a very promising approach. So we were thinking whether we could use a pharmacological agent that we could basically inhibit the crosstalk between the tumor cells and macrophages and thus affect the growth of tumor cells. One of the most potent chemopreventive agents for colorectal cancer is actually vitamin D. Vitamin D deficiency or vitamin D insufficiency is actually associated with significantly increased risk of colorectal cancer. And for us, it was puzzling that actually tumor cells do not respond to vitamin D. Nothing happens if you put vitamin D on tumor cells, although they have functional vitamin D receptor. But in contrast, actually, macrophages were very responsive to vitamin D. They would stop proliferate, they would differentiate. Morphologically, they are quite different. These are untreated macrophages. These are treated, actually, macrophages with vitamin D. And you can see quite clearly that they appear bigger. The ratio between nucleus in blue and the cytoplasm is quite altered. And when we treated our cultures with vitamin D, you can see that the ability of macrophages to promote growth of tumor cells was truly abrogated. Vitamin D inhibited the ability of macrophages to promote Wnt signaling. And vitamin D-treated macrophages actually behave very much like macrophages that lack interleukin-1 gene. You can see here, macrophages with silence interleukin-1 are not good inactivators of JSK3 beta. They do not induce c-Myc expression. And so are vitamin D-treated macrophages. So we tested whether vitamin D actually possibly affects communication between the two cell type by affecting the interleukin-1 synthesis. And it's true, you can see that vitamin D actually is a very potent inhibitor of interleukin-1 B secretion in our co-culture system. And because we showed that STAT1 is the transcription factor that is required for the proper crosstalk between macrophages and tumor cells, We examined whether STAT1 activity actually is affected by vitamin D, and as you can see here, this is antibody that recognizes activated STAT1, that is nuclear STAT1, and in the presence of vitamin D, there is actually no nuclear STAT1. The total level of STAT1 actually is not altered. And by immunofluorescence, you can see that while in untreated macrophages, most of STAT1 indicated in green here would be in the cytoplasm, would be in the nucleus, but vitamin D3 actually inhibits nuclear localization and therefore inhibits transcriptional activity of STAT1. Now, Wnt signaling actually was a new signaling pathway for interleukin-1. So for us, we wanted to understand a little bit how interleukin-1 beta is able to induce Wnt signaling. We worked out the pathway, and I will not talk about that, just to say that an interesting thing was that we showed that interleukin-1 beta signals through pathways such as NF-kappa B and AKT. And these are pathways that are not just growth promoting, but they also promote survival of tumor cells. And we decided to test whether the presence of macrophages then would alter the response of tumor cells to therapeutic agents. And we were interested in TRAIL. TRAIL is a TNF family member. It's an important component of tumor immunosurveillance. And it's a promising therapeutic agent because it has a remarkable selectivity for tumor cells. It induces apoptosis, it kills tumor cells, but doesn't do much harm to normal cells. So we culture a panel of TRAIL-responsive tumor cells in the absence or in the presence of macrophages and tested their responsiveness to TRAIL. And as you can see, every cell line that we tested loses the responsiveness to TRAIL when cultured in the presence of macrophages. This is the extent of apoptosis when we culture cells alone. This is the response, apoptotic response of tumor cells when they are cultured in the presence of monocyte. And we confirmed that biochemically, macrophages inhibit the caspase cascade in tumor cells. Activation of caspase 8, activation of caspase 9 were all inhibited by macrophages. So macrophages clearly secreted a factor that interfered with TRAIL-induced apoptosis. You can see here a picture of the cells. These are normal, healthy cells. These are TRAIL-treated cells. And these are TRAIL-treated cells together with interleukin-1. And these are TRAIL-treated cells cultured in the presence of macrophages. And we show that interleukin-1 is not only sufficient to protect cells from TRAIL-induced apoptosis, but was also required when we silence interleukin-1 in macrophages, when we use neutralizing antibodies specific for interleukin-1, or when we silence STAT1, transcription factor that mediates the crosstalk between tumor and stromal compartment, macrophage is no longer protected from TRAIL-induced apoptosis. This is simply a quantification of the results that I just showed you on the picture. So if interleukin-1 is so critical for the ability of macrophages to interfere with TRAIL-induced apoptosis, then vitamin D, which was a potent inducer of IL-1 production, should do the same. And that's actually exactly what happened. I will just talk about this panel. Again, we measured the apoptotic response of tumor cells to TRAIL. Good amount of apoptosis. Macrophages, very potent inhibitors of apoptosis. And this apoptosis actually was completely restored by addition of vitamin D to the cultures. If we silence vitamin D receptor on tumor cells, the response was not different. But if we silence vitamin D receptor on macrophages, vitamin D did no longer actually did not modulate apoptosis in this cell, confirming that actually vitamin D acts through macrophages, not through tumor cells. We did not know how macrophages protect from trail-induced apoptosis. We tested a couple of usual suspects, but did not found a protein that would be secreted from macrophages and inhibited trail-induced apoptosis. But one thing we knew, and that is that the factor that protects cells from trail-induced apoptosis is a Wnt target gene, because Wnt signaling in tumor cells was required for that. We transfected cells with dominant negative TCF4. This is a dominant negative inhibitor of Wnt signaling. Or with an empty plasmid, empty piece of DNA. And as you can see here, in blue is apoptotic fraction of cells. These are results of flow cytometry. Trail, potent inducer of apoptosis, macrophages or recombinant interleukin-1 would inhibit the apoptotic response, but only in cells that have intact Wnt signaling. Inhibition of Wnt signaling, you can see the macrophages or interleukin-1 no longer protect the cells from trail-induced apoptosis. And we found that the Wnt target gene called SNAIL behaves in a similar pattern. SNAIL is again a DNA binding protein, a transcription factor, but unlike beta-catenin or STAT1, actually, it's usually a transcriptional inhibitor. It plays a very important role in metastasis of cells, and we show that tumor cells cultured in the presence of macrophages or tumor cells treated with interleukin-1 really had elevated levels of SNAIL, and inhibition of Wnt signaling actually prevented increase in SNAIL expression of these cells. And we showed that macrophages stabilize cells, stabilize SNAIL in tumor cells through interleukin-1, because inhibition of interleukin-1 in tumor cells or inhibition of STAT1 once again, or treatment with vitamin D3 actually interfere with SNAIL stabilization in tumor cells. So this is a summary of what I've told you thus far. We showed that macrophages secrete interleukin-1 beta, which in NF-kappa B dependent manner promotes Wnt signaling, and that one of the important mediators between macrophages and tumor cells appears to be SNAIL. What we really didn't know is how tumor cells signal to macrophages for the IL-1 beta response. And maybe we can talk about that in discussion, but a good candidate for that was versicane. Versicane is an extracellular matrix protein. It's a Wnt target gene. If you induce Wnt signaling in tumor cells, you induce versicane. And as you can see here, indicated in red, most tumor cells, most human cancer, the exception is thyroid cancer, have significantly elevated levels of versicane. Red is elevated expression. And because its Wnt target gene is not surprisingly highly elevated in colon cancer cells and in primary colon cancer tumors. To establish whether versicane is indeed a protein that is required for the interaction between tumor cells and macrophages, we silence bursicane in tumor cells. And we first examine whether the levels of interleukin-1 beta are altered. And as you can see here, in contrast to parental cells, tumor cells with no bursicane, with silenced bursicane, were not able to induce secretion of interleukin-1 beta. Here is just shown that cells transfected with bursicane siRNA indeed do not express bursicane at all. And because interleukin-1 was not produced in bursicane transfected tumor cells, Wnt signaling was not induced in bursicane deficient cells, and bursicane deficient cells actually were not protected by macrophages from trail-induced apoptosis. So increased Wnt signaling in parental cells, no increase when we silence bursicane. Good protection from trail-induced apoptosis, and really no protection from trail-induced apoptosis when you silence bursicane. Finally, we show, and that was quite exciting for us, that snail is actually required for the expression of bursicane. We show that overexpression, if we overexpress snail in tumor cells, transcriptional activity of bursicane increases quite a bit. If we silence snail in tumor cells, the levels of bursicane transcription really drops dramatically. And more importantly, actually, this is immunostaining for bursicane. You see, if you silence snail in tumor cells, actually there is no bursicane. It's as good as if you silence bursicane itself. The levels of beta-catenin actually were not affected. And because snail-deficient cells lacked bursicane, snail-deficient cells actually did not stimulate macrophages to produce interleukin-1 beta, as shown here. So a good amount of interleukin-1 induced by parental cells, no response when we use cells with silent snail expression. And likewise, snail-deficient cells were not protected by macrophages from trail-induced apoptosis. To confirm that the interplay between snail and bursicane operates in vivo, actually, we examine Oncomine. This is a publicly available database. It's a collection of microarrays. And as you can see here, the tumors that have low level of snail, in white would be low, in red would be high. Low level of snail always translate to low level of bursicane. High level of snail would translate into high levels of bursicane. So if I summarize, what I told you today is that tumor cells, colon cancer cells in particular, secrete bursicane, which through unknown mechanisms stimulate macrophages, which secrete interleukin-1 beta. Interleukin-1 beta, through receptor binding, activates oncogenic signaling, NF-kappa B, AKT signaling, and promotes Wnt signaling in tumor cells, resulting in induction of genes such as c-Myc, which probably promotes growth of tumor cells. Snail is required for motility of cells, plays an important role in metastasis. And we show that actually it feedbacks to Wnt signaling. Snail is target of Wnt signaling, but also regulates Wnt signaling. And it's required for the expression of bursicane, and it's required from protection of tumor cells from trail-induced apoptosis. So it's really a kind of a self amplifying loop that promotes growth of tumor cells and promotes their survival. We haven't studied metastasis, but the increased level of snail in tumor cells would suggest that macrophages are pro-metastatic, and that has been suggested before. Importantly, actually, we show that we can short-circuit this loop, if you will, that we can interrupt this loop by vitamin D, which binds to macrophages, inhibits activation of STAT1 in macrophages, and therefore inhibits the release of interleukin-1 beta. And this is probably an important slide. Pawan was a postdoc in my lab. She did a lot of work. This is a noob. She didn't do any experiment, but was the best result that Pawan got in my lab, she always said. Jack and Lorianne were involved in the work I didn't talk about today. Jose was a graduate student from Spain. Malika was an MD fellow in the lab. Vincent was a summer student. And in yellow are my collaborators. Takehi and Senji actually supplied us with a lot of reagents that we need for our work, and Sylvie and Michelle supplied us some tissue sections actually that again I didn't have to talk about today. And Len actually is my collaborator, a long-time collaborator at the Albert Einstein Cancer Center. Thank you very much.
So thank you very much, Lydia, for this presentation. That was maybe very specific, but very also effective.
I apologize to all architects and physicists in the audience.
Maybe we can see.
Okay, you want me to sit here? Okay, let me get mine.
So maybe I can start first with some questions, then maybe the audience will join as well, also maybe with curiosity about this specific cancer or general cancer. So, in fact, the first question is how much the mechanisms that you proposed are specific for colorectal cancer, and how instead can maybe be generalized also to other type of cancers?
I don't know how general it is, because you never know until you do the experiment. You know, the fact that macrophages promote tumor growth through secretion of soluble factors is a well-established fact. And it's known for breast cancer, for pancreatic cancer, for other types of cancers. Whether it's always interleukin-1 beta that does the job, we don't know. We simply didn't do that experiment. We haven't tested the system in other cell types. But the general mechanism that it's a tumor-promoting microenvironment, and that macrophages, when recruited to the tumor microenvironment, can promote growth and survival and metastasis, probably would apply to other cancers.
Mm-hmm. Does mean that eliminating macrophages or using anti-inflammatory therapies can have some positive prognosis for cancer progression, or which is the state of the art of the anti-inflammatory use in cancer therapies?
They've been used for decades, probably, in patients who are at high risk for colorectal cancer. You know, aspirin a day takes a doctor away, so they say. So I think macrophages, I'm sure, an important target. You know, an important target of non-steroidal anti-inflammatory agents, which are used therapeutically for patients who are at elevated risk for colon cancer or with familial history of colon cancer. It's used. And the target of these drugs is cyclooxygenase 2. And cyclooxygenase 2 actually is very often not expressed in tumor cells, but it's expressed in macrophages. So I think macrophages are probably physiological targets of these drugs.
And so what you suggest as the most promising target for future therapies?
Oh, that's a difficult question. I don't know. You know, what's known is that cancer is not all cancer cells are equal in tumor, and there is a cell in the tumor called cancer stem cell. Okay? This is the cell that probably will be a target for therapies. It's unfortunately also a cell that is most resistant to therapeutic approaches. But this is like regular stem cells, cancer stem cell actually is the cell that is responsible for the propagation in tumors. So approaches to target the cell specifically are not easy because we do not understand fully the characteristic of cancer stem cells. But there are now several markers that are known to be specific for cancer stem cells. So I think probably we'll have to learn more about the cancer stem cell, and this will be a good target because this is probably the cell that we will have to destroy in order to eliminate cancer.
Mm-hmm. And another question about personalized medicine. Now it's quite well known that each individual genetic repertoire as well as the inflammatory potential is quite variable from person to person. So there are a lot of efforts in research searching for biomarkers and that can give an evaluation of the potential for cancer progression in each person. So do you think that you can comment maybe on personalized medicine strategies in this type of cancer?
Yeah, unfortunately, I would say we are not very close to personalized medicine. Of course, it's a targeted medicine, and personalized medicine, it's probably the medicine of the future, but I don't think we are very close. A typical cancer cell has like 10,000, 20,000 mutations, and we do not understand which mutations are the drivers. We don't understand actually how different a tumor in person A is from a person in tumor B. There have been attempts, you know, people are doing a lot of gene profiling and they try to find characteristics that would help physicians to guide therapies. In colon cancer, therapy is very standardized. Recently, actually, it's known, and that is really known from profiling, that patients who have KRAS mutations, this is a common the oncogenic mutation in colon cancer. For example, they exclude these patients from certain therapies because they know that they would not respond to that. I should also say that I truly believe that tumors in person A and tumors in person B are more different, that it's microenvironment in person A and in person B. Saying that, I believe maybe we do have to target altered tumor microenvironment. A big advantage is, of course, that cells in tumor microenvironments, with rare exceptions, are genetically stable. You don't find mutations in tumor microenvironments. These cells are activated. They secrete interleukin-1, they secrete chemokines, and I think we probably have better tools to deal with that than we have to repair genetic changes in cells.
Okay. So maybe other questions from the audience can be done now, if there are.
This will be a very non-expert question, but I was always under the impression that cancer is a result of a random mutation, so that different cancers are possibly, I mean, each cancer is somewhat different, but then you suggest that there are very specific mechanisms for, let's say, colon cancer, which are, I don't know, the same from person to person. Can you perhaps comment a bit more on that, this, let's say, universal mechanism for colon cancers, which I would imagine are different from person to person because mutations are random?
It's very different, probably. I think they recently sequenced primary tumor from lung cancer patient and I believe breast cancer patient, and I think the number of mutations was 10,000, you know, huge number. Most mutations, of course, are silent. Most mutations are in the regions that do not result in altered growth or in altered survival. What is known is, for example, that in colon cancer, there are certain mutations that the mutation in APC would occur in every patient virtually, almost every patient. If you don't have mutated APC, you would have mutated beta-catenin, which result in virtually the same phenotype. But some changes, obviously, are quite common in all patients.
Other questions? Or maybe, yes.
Another very non-expert question. If I understood correctly, the vitamin D3 suppresses growth of cancer, right?
Yes.
By canceling the feedback mechanism of the process you mentioned. What happens if a person, for example, stops taking D3? Does the proliferation of the cancer appear at a higher rate than before? Does it not reappear? What happens?
I don't think anybody tested that, actually. As I said, vitamin D is what is known, for example, that people with low levels of vitamin D, I don't know actually how it is in Slovenia, in the Bronx where I live, it's like 50%, 60% of populations that is vitamin D deficient, that there is significantly elevated risk of colorectal cancer. And we all take vitamin D pill in the morning for a variety of reasons. Better yet, come to Piran and spend an hour in the sun, and you raise your level vitamin D to the levels that are certainly protected.
But is it possible just to have some statistics of the occurrence of this type of cancer in different regions? Like, for example, in tropical regions, there should be much less than. Were there any statistics made about this?
I suggest that moving to Sweden. Yes. Northern parts of the hemisphere have significantly higher levels of colorectal cancer, actually, and breast cancer. They do. The studies have been done, yeah.
And now another question. I've read somewhere that we have many, it's not connected to this type of research in cancer, but still, we have many more bacteria inside our bodies than our own cells. Was there any research done in using this bacteria to kill the cancer cells in our bodies?
Not so much to kill bacteria. Actually, this question is quite relevant for colorectal cancer because inflammatory bowel disease, which is in parts a high risk for development of colorectal cancer, is inflammation of the intestinal tract, virtually. This disease, the etiology is not quite understood, but appears to be an abnormal response to bacterial flora, and you pointed that rightly. We are made of bacteria, basically. What they try is actually, they try to understand whether patients with colorectal cancer, and this bacteria is now actually quite defined. There are big groups who are studying bacterial flora in the gut. And they're trying to understand whether a certain type of bacteria actually is associated with the disease, meaning whether a colorectal cancer patient would have different composition of bacterial flora than a healthy individual.
Maybe not so cancer related, also normal cell related. I was always interested in how is the speed, or when do the cells know when to stop growing? How is this process done? I mean, in cancer it's probably different. I don't know how long it develops. When does it stop? It's probably different than normal cells.
Yeah.
But what is the process behind this?
You know, that's a characteristic of tumor cells, that it doesn't know when it's time to stop dividing, when it's time to stop growing. Normal cells would grow when you add growth factors, right? When you add a growth factor A or B or whatever the cell needs for proliferation, when you remove growth factor, the cell would stop proliferating. Cancer cell doesn't do that. You know, we all know in the lab, you know, normal cell, there is something called contact inhibition. You know, when you touch a neighboring cell, you stop dividing. Tumor cells basically keep piling on each other. So it's a response to growth factor that is altered. Cancer cells do not know how to respond to factors that normally inhibit the growth of tumor cells. So this response is altered, yeah. You know, I was talking about oncogenic signaling today, Wnt signaling, NF-kappa B signaling. These are signaling pathways that are functional in normal cells, but in contrast to tumor cells, you know, in normal cells, this is transient, you know. Wnt signaling goes up, and it's actually Wnt signaling is required for the stem cell maintenance, but then it drops. Tumor cells maintain the signaling on a certain level, on a high level.
Can you tell us, is there an increased number of macrophages in colon cancer in comparison to normal mucosa? This is the first question. And the second one, what is the mechanism? If it is so, because macrophages, as far as I know, they don't proliferate. Mm-hmm. And the third one, you didn't mention nothing about antigen-presenting cells, mm-hmm. like macrophages are.
Yeah. To answer your first question, some colon cancers have high level of macrophage infiltration, some have none. Some have little, so there is a variation. And in cancer, such as breast cancer, the high level of macrophage infiltration would mean bad prognosis, would mean that this patient would progress. There's a high likelihood that you would find metastasis. In colon cancer, it's a little bit more complicated. There are some studies suggesting that the patients where you find a lot of macrophages would have better, and then there are some studies actually that find just the opposite. Tumor-associated macrophages are very plastic. You know, so it's not just the number of macrophages that you find in tumor that would tell you whether this is good or bad prognosis. It really depends on what type of macrophages you have. And it's not just M1 versus M2 macrophages. It's really quite complicated. So I think the number will not basically be very helpful. Probably we'll have to understand a little bit more about the nature of these macrophages. For example, macrophages that you find in metastasis would be a little bit different than macrophages that you find in the primary tumors. How do macrophages get to tumors? Tumor-associated macrophages are derived from peripheral blood monocytes. that are recruited to tumor microenvironment by a variety of factors produced by tumors. We are studying whether versicane actually is one of these factors that potentially could help to recruit macrophages. And the third question was antigen presenting ability. I think tumors have really learned to basically inhibit the ability of macrophages to present the antigen. So tumor-associated macrophages have a very poor antigen presenting ability.
Well, I think that if there are no other questions, I thank again the invited speaker of this evening, and I invite you everybody to join the buffet and the drinks of the evenings. So thank you again.
Thank you.